Hello everyone, today we will study about one of the viruses, that is poliovirus. So as the name itself suggests, poliovirus causes a disease called as polio, or it is also called as poliomyelitis. So poliomyelitis is a highly infectious disease which is mainly seen in children, and it is caused by poliovirus. So this is characterized by acute flaccid paralysis. So you can see in this picture there will be a paralysis of the involved muscle, and also there will be involvement of the central nervous system. So in poliomyelitis there will be involvement of central nervous system as well as there will be paralysis of the involved muscle. And this poliovirus belongs to the family of Picornaviridae. Under Picornaviridae, it belongs to enteroviruses. So this poliovirus is classified under human enterovirus C group. And poliovirus is having three types of strains. So this virus is classified into three types: poliovirus type 1, poliovirus type 2, poliovirus type 3. So poliovirus is the most commonly seen as epidemics. Poliovirus type 2 is most antigenic, and also it is most easily eradicable. And poliovirus type 3, it can be associated with the vaccine associated paralytic poliomyelitis, so which is a serious condition. Very rarely this can be seen. So these three types of strains of poliovirus are used for the preparation of vaccines, right? So those strains will be attenuated, they will be inactivated. Even though if they are inactivated, very rarely, 1 in million chances that this virus vaccination strain only can cause the paralysis in the person who is taking the vaccination. And coming to morphology of poliovirus, you can see the structure of poliovirus in the diagram. It is a very small sized virus which measures about 27 to 30 nanometer in diameter, and it has a round or spherical shape. And you can see the symmetry of nucleocapsid, which is having icosahedral symmetry. And another important point is it is a non-enveloped virus. So poliovirus is a non-enveloped virus, and it has a nucleocapsid which is having icosahedral symmetry, and capsid is made up of number of capsomers. So each capsomer consists of 4 viral proteins: viral VP1, VP2, VP3 and VP4. And this poliovirus is having a single-stranded RNA as a nucleic acid. This is about the structure of poliovirus. Having spherical shape, measures about 27 to 30 nanometer in diameter, and the nucleocapsid is icosahedral in symmetry, and capsid is composed of 60 subunits or capsomers, which is consisting of 4 viral proteins each. And this poliovirus is having positive sense linear single-stranded RNA. And coming to pathogenesis. So one of the important point is polio infection. It is only seen in humans. So it is only seen in humans, and there will not be any carriers. Only it can affect, so there will be only the cases. There will not be any carriers of polio infection, and polio infects only the humans, and males are more susceptible compared to females. And also it can be commonly seen in children. And coming to mode of transmission, how the poliovirus spread from one person to another person. The most common mode is fecal-oral route, by ingestion of contaminated food or water. Then also it can transmit through respiratory droplets. So if a person is having infection with the poliovirus and another healthy person comes in contact with that person, while talking, coughing or sneezing, the respiratory droplets of the infected person can spread to the healthy person, and also result in infection in that person. And also infection can transmit through conjunctival contact also, rarely. Next, what is the pathogenesis of this polio? How the polio infection spread to the central nervous system? So, I already told that infection most commonly enters, or virus enters through the fecal-oral route or inhalation, right? So, following ingestion of food or inhalation of the respiratory droplets, virus enters into the body. Initially, if the virus is inhaled, it enters into the lymphoid tissues of the tonsils. If it is ingested, it enters into the Peyer's patches in the small intestine and multiplies there. From the lymphoid tissue, it spread to the regional lymph nodes, causing primary viremia. So, initially it is ingested or inhaled, enters into the lymphoid tissues of tonsils or Peyer's patches in the small intestine, multiplies there, then it spreads to regional lymph nodes, maybe cervical lymph node or mesenteric lymph node, and enters the bloodstream, which causes primary viremia. So, viremia means presence of virus in the blood. And through the blood, it spreads to various tissues, mainly involving central nervous system, and multiplies there. And it destroys the anterior horn cells of the spinal cord. And from the spinal cord, again it enters into the blood, causing secondary viremia. Then finally it results in the symptoms like muscle weakness and flaccid paralysis. This is about the pathogenesis of polio. So about clinical features, lab diagnosis and prophylaxis of polio, we can study in the next class. Thank you.